Hello everyone, my name is Jackson Footit, and today we're going to be talking about the dietary patterns and guidelines, the synthesis, assessment, and limitations thereof. First, we're going to start out with a little game plan. We're going to talk about the definitions of a dietary pattern and guideline. Then we're going to talk about the synthesis and assessment of dietary patterns and ultimately guidelines. We're going to go over the three big ways to synthesize and assess these patterns, hypothesis-driven, data-driven, and hybrid methods, and of course their limitations. We'll talk about evidence sources, specifically with experimental and observational data, and then we'll get into the observational data, talk about cohort studies, case control studies, cross-sectional studies, and limitations of those all, and then we'll round it out with a conclusion. We're going to start out with some definitions. So for a dietary pattern, that is the quantities, proportions, variety, or combination of foods and drinks typically consumed, or it can be considered the combination of foods and beverages that constitutes an individual's complete dietary intake over time. And together, these foods and their interactions will produce synergistic health effects. Overall, the emphasis is on the quality and quantity of foods that are consumed habitually with their associated beverage counterparts. Dietary guidelines can be defined as the science-based recommendations providing advice on foods, beverages, and dietary patterns to promote health, meet nutrient needs, and prevent chronic diseases. So now we're going to talk about dietary pattern synthesis and assessment. And the synthesis of dietary patterns starts with evolutionary origins. Some of the key developments in the evolution of humanity and the progression of our society, such as shifting to a meat-based diet, fire, cooking information techniques, and of course the domestication of plants and animals, really significantly influenced our anatomy, our physiology, and our relationship with what we eat. As far as modern scientific analysis to synthesize and assess dietary patterns, there are three main methods available. There are hypothesis-driven methods, there are data-driven methods, and of course hybrid methods. We will talk about all these in greater detail in the coming slides. So we'll start with hypothesis-driven methods and of course the limitations. So a hypothesis-driven method assesses adherence to a predefined dietary pattern using an index or score to define the guidelines that are aligned with our current knowledge and understanding of how those recommendations affect health. There are several patterns that have been discovered using this technique. The Healthy Eating Index, the Mediterranean Diet Score, and the DASH diet, all stemming from the idea that was inspired using the current knowledge base with a goal in mind to ultimately use these dietary patterns to improve health. The limitations of the hypothesis-driven method is that there is subjectivity. There's a focus on specific select types and amounts of dietary constituents. There's an issue with correlations between the different components of a dietary pattern, and there's concerns with the categorization and adherence to specific indexes or scores in whatever analysis, and that could be challenging. Next, we have data-driven methods and limitations. So the dietary intake patterns from a population data through reduction techniques, like the existing data collected from food frequency questionnaires, the recalls, the dietary records, obtain dietary patterns instead of defined guidelines. So we're not starting with anything specifically with an idea, with a hypothesis, with a goal in mind. We're essentially just observing and collecting and analyzing the data through factorial, principal component, and cluster analysis. And we'll talk about those in a little bit, but the Western food pattern, the prudent food pattern were both discovered using this method. And the limitations of this method is the idea that the process relies heavily on subjective choices made by the researcher. So we're not directly hypothesizing an end goal, but what we choose to analyze is very subjective. And there's a limited ability to reproduce and compare the results of a data-driven method, which can kind of have issues, but the final limitation is that there's a lack of health effect interpretation. We're not necessarily interpreting, like a hypothesis-driven method, what we find to a goal. There's no attributing that. It's more of just taking this data and filtering it to see what comes up. Then we have the hybrid approach, which is unsurprisingly a combination of the hypothesis and data-driven methods to identify dietary patterns. There's the reduced rank regression technique, the treelet transformation, and other interventions in which participants are assigned to consume specific dietary patterns. So the reduced rank regression essentially selects a set of disease-related variables, and based on the hypothesized knowledge, derives dietary patterns based on the data available. So it's kind of utilizing that hypothesis-driven approach. And then the treelet transformation produces patterns involving only natural group subsets of the original variables from quantitative pattern extraction and cluster analysis, more of a use of that data-driven approach. Overall, there hasn't been a lot of well-known dietary patterns that stem from using this technique, but the cardiovascular risk-related pattern or the type 2 diabetes risk pattern were both elucidated using this method. So limitations are that there's a high cost and resources and time as well. There's complex data integration, there's an issue with participant adherence, and there's a methodological originality issue as well. So the evidence that is analyzed through the previous three mentioned approaches is based on different trials, and this is a slide that will help kind of explain the sources of evidence and their hierarchy as far as strength. So experimental and observational trials are what provide the evidence base to be analyzed and to create food patterns, dietary guidelines. And so analytic studies can be split up into experimental studies, which famously would be a randomized controlled trial, which is, as you could see, ranking pretty highly as far as strongest scientific evidence. And then we have observational studies, which make up a bulk of nutritional data, food pattern, and dietary guideline synthesis and assessment. These are going to be the cohort studies, the case control studies, cross-sectional studies, and while they're not as scientifically strong as randomized control trials, they still provide quite a bit of information that can help guide professionals to develop patterns and guidelines. Meta-analyses and systematic reviews are unsurprisingly at the top, at the strongest evidence. Randomized control trials, that experimental trial, is right underneath, and then we have our observational trials as well. So let's talk about the experimental trials, the experimental data. As mentioned, the randomized control trials are one of the preferred methods of getting this information. They are great because they are blinding to limit the observer and participant bias and establish causation. However, they might be resource intensive and limited by ethics. It's really hard to gather nutritional data based on randomized control trials because, as we've mentioned in class, people eat foods, they eat combinations of foods, substitutes for within their food groups, so it's really hard to get good data using RCTs. The limitations are, as I just mentioned, as well as the long time it may take to finish the trials and the resulting evidence gaps thereof. As far as experimental trials that deal with nutritional evidence, dietary control trials are great because they can determine causal relationships and act as hallmark studies for the development of patterns and guidelines. These are trials that modify dietary intakes or habits and evaluate their potential effects on outcomes to determine causal relationships, which provides pretty strong evidence. Their limitations are, of course, their complexity in nature. the collinearity between diet compounds, the multi-target effects of the interventions, diversity in dietary behaviors, food culture, dietary status, control groups, randomization, and of course adherence, which will be a consistent issue throughout these studies. So next we have observational data, the observational studies. These consist of cohort studies, case control studies, and cross-sectional studies. There are others, but they're not really relevant to nutrition research. So the case control and cohort studies are able to causally measure disease occurrence and its association with an exposure. It's not entirely causal. It depends, greatly on how the study is set up and what it has to offer, but they are a bit more useful than cross-sectional studies because cross-sectional studies examine the data on disease and exposure at one particular point in time. So because of the relationship between the disease occurrence and the exposure cannot be established, these cross-sectional studies don't really have a cause and effect relationship that we can see in cohort or case control studies. But as I mentioned earlier, the observational studies, especially the cohort studies, make up a huge bulk of nutritional evidence that's available and the basis for a lot of interventions and recommendations. So we have cohort studies. As far as the method and limitations, cohort studies are those that track groups over a long period of time to evaluate results. So the prospective cohort studies are what make up a bulk of cohort and observational studies used. They follow groups of healthy individuals forward in time from the present, tracking their dietary and lifestyle habits to see the developed diseases or outcomes. The limitations of these kinds of studies is the long follow-up period, potential lurking or hidden variables, errors with measurement, biases, and a notable, just modest disease-diet relationship. So next we have case control studies, which are studies that identify subjects by a certain outcome at the beginning and then compare those with a medical condition to those without. So outcome status is confirmed and subjects are then categorized as cases, controls are selected, and data on an exposure to a risk factor or several risk factors are then collected retrospectively. The limitations of these studies would be a difficulty selecting similar participants and a dependence on participant memory. This, of course, can lead to bias and incomplete data sets. And then we have cross-sectional studies. So these studies measure the outcome and the exposures in the study participants that are selected at random at the same time. An example of this would be a survey. It's inform the prevalence of a particular disease, but it's not providing information on causality or what the best treatment might be. So these limitations would be a single measurement of an exposure or outcome. If you take a survey, you're not going to take another one next week and the next week. There is an issue with memory and biases. The prevalence of an outcome depends on the incidence of the disease, and then, of course, the survival of particular participants. I understand this has been a pretty boring presentation, but to wrap it up, we first talked about dietary patterns and guidelines and why they're important for nutrition, education, information, and human health. We talked about the main methods used to synthesize and assess dietary patterns, that hypothesis, data, driven, and hybrid methods. And then we talked about where evidence came from to be analyzed, talked specifically about experimental versus observational data and the strength of those. Right under that, of course, we have the randomized control trials and the dietary control trials, which are stronger evidence than the prospective cohorts, which is slightly stronger than case control, and of course stronger than cross-sectional. So one can see that there is a lot of information, and because there's no great way to determine causality via experimental randomized control trials, we have to rely on these recommendations from observational data, which is good because they can provide a basis for proper patterns and guidelines, but they fail to have the same causality as like a pharmacological trial would. here are my sources that I cited, and thank you very much for sticking around till the end. I can't wait to see everybody else's presentation.